Good afternoon, everyone, and welcome to the 2026 CRB webinar series. Today's webinar will review plant growth regulators currently available to the California citrus industry. I am Caitlin Stanton with the Citrus Research Board, and I'm joined by Megan Iott, who will be introducing our speaker today. We encourage you to ask questions during the webinar using the Q&A function, which can be found at the bottom of your screen. All questions will be answered at the end of the presentation. With that, I'm going to pass it over to Megan, who will introduce our speaker.
Thank you, Caitlin. Please welcome our first speaker, University of California, Riverside Extension subtropical horticulturalist, Dr. Mary Lu Arpaia. Dr. Arpaia conducts research in two areas of subtropical fruit post-harvest handling, the role of pre-harvest management on post-harvest fruit quality, and the response of subtropical fruit to the post-harvest environment, including temperature during handling and ethylene exposure. Today, Dr. Arpaia will discuss plant growth regulators available to the California citrus industry, the effects of each of the PGRs, and the importance of 2,4-D timing with respect to the premature fruit drop observed in fall of 25. So, Dr. Mary Lu Arpaia, please go ahead and share your screen to begin the presentation.
Well, great. Thank you for the kind introduction, Megan, and welcome everybody. Good afternoon to everybody attending the meeting. And my job, as Megan indicated, was to talk a little bit about plant growth regulators, review what we have registered in California, and talk about a couple other little things along the way. So it's a presentation that is going to be sort of moving around a little bit. Okay, so the outline of the talk is I'm going to review plant growth regulators, not in great detail because time doesn't allow that, but for California citrus. I was asked to make a few comments about puff and crease, and as I was telling Melinda and Megan before the meeting, actually, it was a great opportunity for me to review what's been going on in this subject. So I'll talk a little bit about that. And then Tripti Vishwas, who is a counterpart of mine at Lake Alfred at the University of Florida. She kindly shared some recent information that she has on fruit drop, and I'll share a little bit about that. And then at the end, I'll end up on going over the survey that the Citrus Research Board conducted about the premature fruit drop problem that we had earlier this year. So there are multiple types of plant growth regulators out there. And the thing is that we have to remember when we're using these plant growth regulators is that they don't have just one effect on the plant. These are endogenous, the plant growth regulators that we use either trigger or they mimic endogenous plant hormones that are found within the plant. And so these plant hormones have very multiple jobs that they do within the plant. And so dose is always very important. What organ of the plant you're targeting, and also the dosage, and then time is very, very important. And so dosage and timing then become key when you're applying these materials, and also then good spray coverage. And more in the literature, as I reviewed recent literature, there is definitely a lot of mention about having the right climatic conditions either before, during, and after a spray application. So examples of plant growth regulators that are commercially used are auxins, gibberellins, cytokinins, ethylene, and abscisic acid. And so in the citrus world, we have auxins, gibberellins, and ethylene registered for use here in California. So these plant growth hormones, phytohormones, have many different effects. So auxin is a primary regulator within the plant. A lot of research has been done on auxins. It typically, in a nutshell, will promote growth through cell enlargement. So not cell division, but cell enlargement, making the cell grow. It's involved in apical dominance, so it's used commonly to break apical dominance. It's involved in root promotion, fruit thinning, and fruit drop prevention. Gibberellins also promote growth through cell enlargement, but these materials are used to promote fruit set, flower induction, and they can also be used for thinning. In some crops, it can be used for thinning, dormancy break, seed germination, and what we're interested in terms of citrus, delayed senescence. The cytokines, there's been work in citrus on cytokines, and there's recent work actually in Florida looking at cytokines, but now they're different. They promote growth by promoting cell division. So cytokines then promote cell division. They also counteract apical dominance, so they're working sort of against auxin. They're involved in branching, also in senescence, and they can also be involved in fruit abscission. Ethylene we use widely to induce ripening of fruit. It's involved in leaf abscission. A good example of that is the use of ethylene in cotton to promote leaf abscission before the cotton is harvested. It promotes radical growth in roots as well. So it's involved in many, many different things. It can also be used to induce flowering. A good example of that is ethylene sprays in pineapple. So abscisic acid, or ABA, it's considered to be promote plant stress, regulates plant stress. It's involved in the opening and closing of the stomata on leaves. It can also promote leaf and fruit abscission, and it's been found to regulate dormancy in perennial crops. Now, so when I went to graduate school, those were the 5 classes I learned about in plant growth regulators. But since that time, we have newer plant growth regulators that are talked about. So the first one is the jasminates. They're believed to be involved in like a response to damage to the tissue and environmental stress. And you can see that they're involved in processes like root development, pollen maturation, and leaf senescence. Another one that is more of a stress-related plant hormone that's involved in soliciting immune responses is salicylic acid. And there's a lot of work actually in the literature now about salicylic acid and triggering different immune responses in plants. The 2 newer ones, and probably the 2 latest ones to be really described, are the brasinosteroids. They regulate cell division and elongation. They're involved in vascular differentiation and reproductive development. And again, these have been found to have elevated levels when the plant is under stress, especially in terms of drought and extreme temperatures. And then the final one is stringolactones, and they're really interested because they seem to be involved in controlling tree architecture in terms of the shape architecture and the amount of branching that you can get. So that's actually very interesting. And again, you can see environmental stress. So all of these, to me, when I read the reviews about all these different classes of plant growth regulators, is that they have multiple ways that they act within the plant. They all are influenced somehow by environmental stress. We're living in a world now where the climate is less predictable than it was 10, 15 years ago. And so certainly these hormones are going to be involved in how our citrus trees respond when we have environmental stress. So these are the 3 plant growth regulators that are currently registered in California for use. So auxins, gibberellins, and ethylene, and these other ones, there are products out there that could be tested to look at whether it would be worthwhile to register within the state of California. So I don't really want to go much further in talking about plant growth regulators without paying a tribute to Dr. Coggins. Charlie Coggins was a member of the botany department at UC Riverside, and really he is the reason why we have auxins and gibberellins and registered in California for citrus. He was a leader in terms of getting gibberellic acid registered for both pre and post-harvest. He was a reason why we have 2,4-D and NAA, and all that work was done when he was first hired at UC Riverside in the 60s, and he had a very long career into the early 1990s. When he retired in the early 1990s, Carol Lovett picked up and carried on a lot of his work looking at the effect of plant growth regulators. And in fact, it was her work with Thomas Chow that allows us now to have gibberellin registered for fruit set on clementine mandarins. So UC Riverside has a long history working in this area. So we have both pre-harvest and post-harvest applications for these plant growth regulators. In terms of pre-harvest use, we have auxin registered for a couple different uses. The main reason 2,4-D, the isopropyl ester of 2,4-D is registered for pre-harvest fruit drop and fruit sizing. We have NAA. I won't, it's a tongue twister of a name, so I'm just going to say NAA, another synthetic auxin, is used for chemical thinning. And then really a very, very important tool in our chest, especially for navel oranges, is gibberellic acid, because GA3 delays rind aging or the aging of the peel. It reduces the physiological disorders that are associated with rind aging, such as thick, sticky peel or navel rind stain, for instance. It's registered for use in controlling creasing in Valencia oranges, although it does also have an effect on navels and mandarins. And it's registered for use for increasing fruit set and yield for clementines. Now, I know probably most of you listening to this talk are in the field, they're not in post-harvest, but the post-harvest use of these materials is also very important. A very important use is the isopropyl ester of 2,4-D for button retention during the storage of lemons, because that is correlated with the incidence of alternaria stem end rot with long-term storage. Gibberellic acid, this was work done by Dr. Coggins just before he retired. He demonstrated that there was delayed coloring in the storage of the lemons and peel-related issues. And then ethylene, of course, we use ethylene for degreening of citrus with early season fruit. So I wanted to review this. So what you see down in figure B on the left, that is the relative fruit growth for citrus, regardless of whether it's a grapefruit or a mandarin or an orange. And citrus goes through 3 different distinct phases. So we have stage 1, which is really the time of active cell division. Stage 2 is really the time for cell enlargement. And stage 3 is really then once the fruit mature and it begins to senesce on the tree. And so when we think about where we apply the plant growth regulators, we're applying plant growth regulators that target flower abscission, fruit set, and early fruit development. And then we also are targeting then the point at which the fruit is horticulturally, physiologically mature, and it's starting to age on the tree. And so we're applying materials to stop fruit drop and also the aging of the peel. So I'll go through each of these classes briefly. So for auxins, again, we have the isopropyl ester form, a 2,4-D register, as well as NAA. And so there's several uses of 2,4-D in California that are registered. So one of the main important ones is increasing fruit size of navel and Valencia orange. You can see we also have a registration for grapefruit mandarins and mandarin hybrids. The dose that you apply is dependent upon the size of the fruit, but typically when the fruit are still small, you can see for oranges, less than 3/4 of an inch or an inch for grapefruit. The timing in general is 21 to 35 days after 75% petal fall. So if you think about the growth of the curve, you're targeting a time when you apply these materials during that stage 1 growth, during the time of active cell division. And it's believed that the auxin works because part of it is that it is having some effect on thinning, actually, of the crop on the tree that allows the fruit that remained to increase in fruit size, but is also in many ways believed to also, even though auxin doesn't promote cell division, is allowing cell division to occur so that those fruit grow faster. Only one application is allowed per cropping cycle. And I noted on the label that if you apply during periods of low relative humidity and high temperature, just like NAA, you can actually cause thinning of the crop. So that's an important thing to consider when one is applying this material to increase fruit size. And as we all know, like this last spring, we had several days where it was very hot, and that would have, if it had been applied during that period of time, just like NAA, you may have seen a little bit more fruit drop. A second use, and probably the most important use pre-harvest, is for control of fruit drop. And 2,4-D is registered for oranges, grapefruit, lemons, tangelos, and other citrus hybrids and tangerines in California. You're limited to one application. You're not supposed to apply within 7 days of harvest. The label also indicates that you should avoid applying to trees that are less than 6 years old. And the reason they give is that depending on when you apply it, you can affect the growth habit of the tree. The nice thing is that this can be applied in conjunction with jib, so that is very nice. But another caution label on the label is avoid high temperatures. And so when we have high temperatures in the fall and you apply this, you can actually stimulate fruit drop. So the post-harvest use of 2,4-D is for bunting retention for lemons during storage. As I mentioned earlier, if the fruit has a button on it, then applying 2,4-D when the fruit goes through the pack line for prior to storage, that 2,4-D will actually reduce the incidence and severity of alternator stem end rot. And I have personal experience with this because back in the 80s and early 90s, we had to get the isopropyl ester form of 2,4-D re-registered in California. And in conjunction with Irv Eakes and Joe Eckert, we did a very large study looking at manipulating lemons to see if we could replace the 2,4-D and keep the buttons retained. And we could not find anything that worked as well as the 2,4-D. And fortunately, it was re-registered. So the 2,4-D can be applied within the storage wax, or it can be applied following a water rinse before the storage wax application. It has a minimal effect on delaying coloration of the fruit. It's very, very, very minor, and is certainly not as effective as gibberellic acid in delaying the coloration of the fruit in storage. But it does have a very dramatic effect on button retention, even when the button is partially damaged from the harvesting process. So it also, on the label, can be used to reduce leaf and fruit drop when a pesticide oil spray is made. And that can be very important. It can have a limited amount of carryover for controlling pre-harvest fruit drop. And on the label, they specify then, depending on when that pesticide oil spray is used, if you should go back with a mature fruit drop spray. Okay, so if for instance, if that pesticide oil spray with the 2,4-D is made more than 6 months before harvest, then you go back and you can do a full rate application. But you can see if that pesticide application was made within 3 or 4 months of fruit maturation, then depending on the dosage that you use within that oil spray, some of the you would either not go back with that mature fruit spray, or you would apply only about 25% of the additional 2,4-D. So that's an important use, because we all know that with oil sprays you can get leaf, especially a lot of leaf drop. So we'll move on to NAA. It's used as a chemical thinner. And I remember Dr. Coggins sort of shaking his head with me. He goes, I helped get it registered, but I'm really nervous. It can have variable results. It should only be used when you have a heavy crop and you want to reduce some of that heavy crop so that you can increase the size of the fruit remaining on the tree. The big question about using NAA, because we'd be applying it now also during stage 1 growth. So you're applying it during that rapid cell division. You want to apply it. You can see when the fruit are 5 to 20 millimeters in diameter, so quite small. But the problem is that either high temperature before, during, or after the application, you can get drastic overthinning. And so that was the thing that I remember Dr. Coggins telling me, that he was very worried about the registration of this material because he was very worried about people applying it and then we having a heat spell. Like if you had applied it last week or early this week, and now we're going to have temperatures this coming week in the next couple of days well over 100 degrees. That would be a situation where you might end up with overthinning of the crop. So that's something that needs to be considered when using this material. Move on to gibberellic acid. It's involved in peel aging, reducing peel aging, post-harvest storage, and we also have a registration for fruit set increase. So the pre-harvest use, I think everybody's aware of how important this product is for delaying rind aging and reducing physiological disorders of navel orange. Typically, what we do is we apply it about 2 weeks before color break, and that is very successful then in delaying the coloration of the peel. Number one, the coloration of the peel is delayed by, and depending on the year, anywhere from about 3 to 5 weeks. And it also, if you're holding navels later into the season, it's definitely known to reduce issues such as rind staining, water spotting, sticky rind, puff and crease, and peel softening. And so it's a very, very important tool for us. It's also registered to decrease creasing and peel aging of Valencia orange. And in that case, what you're targeting is the young fruit in August to October, applying it for a crop that you're going to harvest 6 to 7 months later. But again, it has to do with the protecting and reducing the aging of the peel. So it's also registered for mandarins and grapefruit and pumelos for the same reason, for puffiness, rind peeling, softening. And it's also been shown to have some effect on reducing pre-harvest drop of grapefruit. That's in the literature. So just to summarize, because gibberellic acid is really, really important, it delays color break. The rind senescence issue is very, very important. It helps the fruit retain the firmness of the peel, which is very important. We don't want to have soft peels. It reduces navel rind strain on years where that is a problem. It doesn't solve the puff and crease problem, but what it does, it reduces puff and crease, because puff and crease is a fruit senescent problem. And so by delaying the senescence of the peel, you're reducing puff and crease. It has a carryover effect into the post-harvest world because it reduces susceptibility to chilling injury. And that was brought home to me in particular when I was working on the McCullough project many years ago, where in that project they were looking at many different things: irrigation, nitrogen fertilization, the use of Gib 2,4-D, and many other factors. And Dr. Eakes and I were responsible for looking at the post-harvest storage of the fruit. And we had we did storage work for 3 years. And what we found in that was that when we had that that pre-color break application, when we harvested the fruit, the trial was done with, if I remember right, Parent Washington or Fisher. I don't remember which one, but it was one of the two. And when we harvested the fruit in January and we put the fruit into cold storage, especially low temperature, something like that would be in for a quarantine treatment. With the January harvest, the fruit was still very strong, and we saw no difference between fruit that had been jibbed or not jibbed in terms of development of chilling injury. But when we postponed that harvest until March, at the end of the harvest season, there was a very strong carryover effect that in all 3 years, the fruit that had been treated with gibberellin prior to harvest at the color break time, that fruit withstood in March that very low temperature storage treatment, and actually a typical storage of the fruit. So it did have a carryover effect even 6-7 months later when the fruit went into storage. Work that Mohammed El-Atmani did under Dr. Coggins, they looked at what happens to the surface of the fruit by the jib application. And I was going to put pictures in, but it's sort of complicated. But basically what I can tell you is that the use of gibberellic acid at that color break treatment, if you look at the surface of the fruit, there's natural wax platelets on the surface of the fruit. That's a natural wax coating on the fruit. And when the fruit is immature, that wax is like plates. There's all these different plates of wax. The wax is organized in platelets. However, when the fruit ages on the tree, that wax over time between the time of color break to the time the fruit is harvested in February and March, because they looked at parent Washington, they saw that in untreated fruit, that wax just basically has a melted appearance. And they looked then at what happens when you apply jib, and what happens is the fruit that were treated with gibberellic acid, that wax remained, that platelet organization of the epicuticular wax was retained into the platelets. They also looked at navel rind stain because, again, this is associated with gibberellin. And indeed, again, they saw that in areas of the fruit which were susceptible to navel rind stain, that the wax structure was, the composition of the wax also was affected. So this is just some work from Dr. Coggins that was done in the late 60s, early 70s, looking at the effect of a pre-harvest application prior to color break of gibberellin on the incidence of navel orange rind stain. And you can see it's greatly reduced, and the concentrations we use commercially are based on a lot of his work. This is some data from Spain that the effect of gibberellin on firmness of the peel and aging. So in the top, we have the firmness of the fruit from two different harvests. So the red line, the fruit was harvested in November, and on the blue line, the fruit was harvested in January. So you can see that in that early season harvest, that the inclusion of gibberellin on the tree had a great effect on retaining the peel firmness of the fruit. Less so by the time that you got to the satsuma at the end of the harvest season, but you can see in early November, when we're usually harvesting satsumas, it had a very strong positive effect on the firmness of the peel, and that sort of was curvilinear, maxing out at about 20 parts per million. The lower graph is again comparing the percent ripe fruit in early November. And ripe fruit, by this, they mean fruit that are fully colored. And so you can see, again, the gibberellin in this situation delayed the coloring of the peel. But the red line is the percentage of the fruit that were puffy. And again, you can see that the effect of the gibberellin is very strong on the incidence of puffy fruit. And this was evaluated in January. And again, you can also see that sort of break point of 20 parts per million. So that leads to gibberellic acid in lemons. It can be used in the field to decrease rind aging and yellowing and coloration in both lemons and limes, as registered in California. And here you apply when the fruit are about half to full size, but still full green. So if you want to be able to delay harvest with the fruit on the tree, you would apply it when the fruit are half to full size, but still full green, not fully colored. But importantly now, and this is only since the 1990s, we also will put gibberellin in the storage wax of lemons going into storage. And because just like on the tree, it will delay the chlorophyll breakdown and coloration and maintain the quality of the peel in long-term storage. So this helps us be able to store fruit longer and hold fruit so that we can meet market demands when the market is ready to receive fruit. And you apply this then to the fruit when it is going over the packing line prior to storage. And then in about 15, 20 years ago now, work was done by Thomas Chow and Carol Lovett, looking at the effect of gibberellic acid to increase fruit set and yield in clementine mandarins. And the label says you're allowed to apply 1 to 3 applications from early bloom up to about 4 weeks after petal fall. And they in the publication that they publish, as well as in the label, they really stress that this is a treatment you would use when you're going what is crop load dependent. Okay, so when you're in a low crop yield year, this is when you would want to apply it versus on crop when you have a lot of fruit. Okay, so this is definitely dependent on where you are in alternate bearing of the mandarins. Now, I should say these plant growth regulators are registered for other uses, additional uses in Florida, but they're not registered for use here in California. So I won't review what goes on in Florida. So just strategies to maximize benefit of gibberellic acid. Okay, so previous work by Coggins showed the best time to spray gibberellic acid is late September to mid-October, general rule, 2 weeks prior to color break. This was an important point. This is summarized by an article that Craig Carlson had written a couple of years ago, is that, if you take the time and the trouble to spray the tree with gibberellin to slow down the aging of the fruit, gibberellic acid has no effect on fruit drop. So retaining the fruit on the tree. So if you're going to go down the path of spraying gibberellin, then you definitely need a follow-up or include 2,4-D in the spray when you apply it. Gibberellic acid has the best uptake when the spray is slightly acidic. Obviously, you need good spray coverage. And you can get leaf and fruit drop after a spray with gibberellic acid if you're within 1 or 2 weeks of applying a narrow range oil spray. And again, and this is a common theme. If your trees are stressed, you may get fruit drop with this. You may do other things with gibberellic acid. If you spray too late with gibberellic acid, you can affect the return bloom the following year. And so one of the things that Craig talked about was that once the fruit starts turning color, so you come in prior to color break, when the fruit is sort of yellow-green, orange-green, then you should not be applying gibberellic acid because the gibberellic acid could sort of keep that fruit in suspended animation, so to speak, in that color. If you feel that you want to apply gibberellin again, then you can go in with another spray when the fruit are full color. But the point being is if you wait too long with that spray, Dr. Coggins remember telling me you don't really want to spray gib on the navel oranges, for instance, after December because of the fact that you're having flower induction at that time and gibberellin can affect flower development. So you have to be very, very careful. Ethylene, we have many uses for ethylene. I won't really cover that because they're all post-harvest. So I was asked to say a few words about puff and crease. Another name for puff and crease, a more recent name in the scientific literature now is called albedo breakdown. And why is it called albedo breakdown? Because it's the albedo in the peel, the white portion of the peel, that actually is a contributor to puff and crease. So we'll go back to this growth pattern of sweet oranges. We have the stage 1, the stage 2, and the stage 3. And what's really, really important here is that at the end of stage 1, which is the time of cell division, that is when the peel has its maximum thickness. And you need to remember that one, okay? And so stage 2 is the time of cell enlargement, and stage 3 is really fruit maturation and senescence on the tree. And so puff and crease is a physiological disorder where the spongy tissue of the white tissue of the citrus rind, which is the albedo, separates from the flesh and the outer fruit. And that's shown in this nice picture from Australia. And it's a worldwide problem. That reminds me, it's a worldwide problem. We're not the only ones that have problem. So the literature is very, very confusing and contradictory. So no consistent trends have been outlined. There is a literature review that was published this year. I read it. The literature is confusing. What we do know, fruit maturity, the higher the maturity, the greater the risk, because this is really a senescence issue. As the peel is aging, and that is where you get the problem. We know that the application of gibberellic acid will reduce puff and crease by the fact that it slows the aging of the peel, but it is not a cure for puff and crease. The literature says the fruit with thicker rinds tend to have a lower incidence, but then I found reports that are contradictory to that. Some reports suggest that calcium is important, that calcium uptake into that fruit in the phase 1 growth, because that calcium is involved in pectin development between the cell walls, that if you can get calcium in, you're going to reduce your susceptibility to puff and crease. But there are other reports that say that calcium has no effect. And the other problem with calcium is very, very difficult to get calcium into the plant. John Bauer from South Africa suggested there was a possible role for molybdenum during stage 1 growth, again, that time leading up to maximum peel thickness. Anwar Ali and Carol Lovett highlighted the role of potassium having lower incidence, but then there is stuff in the literature that says you get higher incidence with high potassium. More and more, the reviews I read, weather variability during stage 1 growth is implicated with higher crease and puff later on when the fruit matures. And it is especially true when you have a high temperature fluctuation. So this last spring might be a good year where we might have more puff and crease come this fall on our navel oranges, for instance. One thing that is very clear from the reading the literature, that for puff and crease or albedo breakdown, whatever you want to call it, it is a stage 1. What happens in stage 1 during cell division sets the fruit up to be susceptible or not susceptible when the fruit ages on the tree 7, 8, 9 months later. So that's really, really important. That is at stage 1 growth. Anything that we might want to throw at the fruit, calcium, making sure the potassium is higher in the peel, et cetera, is very important to be targeting the fruit during that stage 1 growth, that time of rapid cell division. So here is potassium, I mentioned that, phosphorus. So again, there's a lot of things in the literature saying thinner peeled fruit are more susceptible. And so this is old data from Embleton and Jones looking at the effect of phosphorus on peel quality or peel thickness of Valencia orange. And then more work that was done by Embleton and Jones. This is from the showing the interaction of nitrogen and phosphorus on a navel orange peel quality. And so we do need to be mindful of what we're doing when we fertilize our plants, that these nutrients interact with each other. So I want to, in the last 10, 15 minutes, I want to go over some data that Tripti Vahesh from the University of Florida at Lake Alfred shared with me on some work that she's been doing looking at 2,4-D and gibberellic acid and fruit drop in Florida. So she did a couple different studies, and all this work has been recently published in 2026. So one question they asked is, can the efficacy of 2,4-D against pre-harvest fruit drop increase instead of doing one dose, they do multiple smaller doses? And can they integrate gibberellin with a labeled rate of 2,4-D to improve pre-harvest fruit drop? So that's be more similar to what we do here in California. And so they did a trial here. You can see they collected 2 years of data. They looked at canopy density, and they had these 4 treatments: untreated control, 2,4-D application in November, 2,4-D broken up into 3 applications, September, October, and November, and then they had the 2,4-D plus the gibberellin with a November application. And of course, we have our problems, and they have hurricanes in Florida, and so they were affected by the Hurricane Milton in the second year. But what they see is it's that mix and the one-time application. They had the lowest fruit drop, and when they applied the 2,4-D in 3 smaller applications, they had slightly higher fruit drop compared to the control. And when they looked at the yield, because they have less fruit drop, especially with the hurricane, they found that the fruit stayed on the tree better even in the hurricane. So that was very good. So they had no differences in yield efficiency, how efficient the tree was in producing fruit in the first year. But in the second year, thanks to the hurricane, they were able to see that fruit stuck on the tree better when they had been treated. And that's yield efficiency there. And they also found that if you just look at the timing, so they did a second trial, and where they looked at September versus November, and you can see here that they definitely had, in this study, lower fruit drop. This is on Hamlin oranges, because the other previous work was done with I can't remember, it was another sweet orange. But when they repeated this work on Hamlin orange, you can see that the application made in September actually had the lowest fruit drop, and they promoted fruit drop when they applied the 2,4-D in November. And actually, they say that they actually had more fruit drop. They had a warm winter that year, and they felt that the application in that November application promoted fruit drop. So the conclusion was, if the label ever opens up in September applications, the Hamlin orange would benefit if you expect the harvest to be delayed, and that but currently they don't have it recommended for the Hamlin orange. But they also did some work with phytohormone analysis of the peel. And actually, they looked at the abscission zone of the citrus as well. And they actually looked at abscisic acid and jasmonal isoleucine, which is part of the jasmonic pathway. And their data suggests there is a hormonal mechanism for fruit drop under stress conditions. And basically what they conclude is that 2,4-D and gibberellin work on separate hormonal pathways, reducing fruit drop, and that gibberellin works by balancing elevated stress hormones that contribute to stress-induced abscission, while 2,4-D increases the auxin within the fruit, and that increase in the free auxin suppresses the abscission signaling, because auxin is antagonistic to abscisic acid. They did another study here looking at that was also recently published, and they did 3 studies. They asked the question in study 1, does combining PGRs provide an additive effect? And that was basically combining gibberellin and a cytokinin and 2,4-D. The other one is the second field study was how can we reduce fruit drop? And the third one was combining strategies to boost production. And I just have 2 slides from the conclusion on this. They conclude that by combining growth promoting plant growth regulators in a phenology base, so depending on what the tree is actually doing, not by calendar, by what the tree is doing, they can improve both the canopy structure of the tree and overall productivity of the tree. That can combine the PGRs with other approaches, including antibiotic yield, trunk injections, and that increases yield. And again, they demonstrate from their work that GA plus 2,4-D can reduce fruit drop. And so especially they say if a hurricane is forecast, is to go and apply this material. So the take-home message is that the combination of PGRs and antibiotics increase yield more than either strategy alone. So the PGRs are not a substitute for the antibiotics. That again, it's an additive approach to approach things by adding things up, not just by replacing. And the right plant growth regulator is all about timing and which one you apply. And again, they're showing what we sort of know that gibberellin and 2,4-D can't combine, can mitigate fruit drop. So that leads to the premature fruit drop issue that we've had in California this last winter. And I want to thank Megan for providing me this slide set. So I've sort of streamlined the slide set that CRB has presented in the past. So CRB did 2 different surveys. So they did one survey where they had 84 respondents. They sent it out to all the growers in California. 52 people actually completed the survey, and they had 92 different grows represented in those 52 responses, a total of 3,700 acres. And the average fruit drop that was reported was 29%, but ranging from 0 to 90%. And they followed that, and here what you can see, this is sort of a summary of that first survey. You can see most of the respondents were from Tulare and Fresno County. The other thing, you can see that 2/3 of the respondents reported on mandarin fruit drop. And in Fresno, about a little bit over half of the respondents responded with, yes, we have a problem, and it's on mandarins. And so you can see here that the kind of fruit drop that is being reported from that survey. So they followed that up with a second survey. They sent the survey to everybody that had responded to the first survey, and on the second survey, they had a 60% response rate. And in this survey, they focused on the timing and whether 2,4-D had been applied, and the timing and the rating and the mixes that have been applied. They also looked at color break, if people knew when color break actually had occurred rather than I think it occurred, because that's what it always says every year. They looked at alternate bearing, the presence or absence of soil-borne pathogens, whether the grower was organic or conventional, and working with others, we looked at weather data. And so they found that it was about split, about half the people applied 2,4-D, the other half did not. And it was applied as a taint mix in all cases. And a little bit over 2/3 of the respondents said that they mixed it with copper lime and a sticker spreader. So the question is, did 2,4-D modify this unusual fruit drop problem we had, especially in mandarins? And that story is a little bit mixed. You can see in Madera County, everybody that responded had applied 2,4-D. But if you look at Fresno and Tulare County, in Fresno County, which is a small sample size, more people that applied 2,4-D had higher fruit drop than those that did not. But Tulare County has the best sample size. And there you can see that the individuals who did not apply 2,4-D reported on average a 36% fruit drop. And in those that applied 2,4-D had a 20% fruit drop. So there is a suggestion here that the 2,4-D application positively impacted the amount of drop that was observed this last fall and winter. So summary and next steps. So we worked, I talked to Megan and I talked to Melinda. They worked very hard on trying to figure all this out. And we went to a climate scientist at UC Merced, and he highlighted sort of like what we knew, that the weather in October and November on average was about 4 degrees higher than normal, especially the nighttime temperatures were higher. And so that warm weather would promote aging of the fruit. Okay. The mandarin was the most effective. We found that out from the survey. There was no link to rootstock. All mandarins seemed to be more or less equally affected that was reported, but most of the fruit that was reported on was W. murcott and tango. And there seems to be a correlation with the 2,4-D application, that those that applied had less fruit drop. So this begs, that maybe we need to be looking at fruit maturation on the trees and the timing of a 2,4-D application. So it could be that, like in Florida, maybe we would benefit from an earlier application of 2,4-D. And so instead of applying it in October to mid-October, like we normally do, maybe going back into September or early September might benefit. But then we also have to think about the fact that if we have spells of very high temperature, we could negatively impact the 2,4-D. Instead of promoting the fruit sticking on the tree, we could actually thin our fruit. So it's something that needs to be approached very carefully and with a lot of thought. So I want to thank you for your attention on this, and I'll turn it over for questions. Thank you.
Thank you, Mary Lou. We are now going to go ahead and ask any questions. If you have any questions for Mary Lou, please use the Q&A function, and we will get those answered. This first question, if the presentation will be available after the webinar, we'll actually have the recorded presentation available on our website.
Yeah, and I can provide the PowerPoint as a PDF to CRB. I'm happy to do that.
Perfect.
Well, I have a question probably to start it off for you, Mary Lou.
Okay.
So I know for the fruit drop we saw this last fall, we saw a lot of moisture, a lot of because of all the tule fog and everything. Was there anything in the literature that also indicated that maybe in addition to like timing and dosing, was there anything that mentioned um, water or amount of moisture or anything that could like exasperate the effects?
That's a great question. I didn't see anything in the literature when I was reviewing it, but this recent graduate thesis from South Africa, they compared, well, they were looking at creasing, but they talk about fruit drop in that thesis. And they compared the Western Cape, which was, Mediterranean climate, to upcountry production they have north of Johannesburg, where it's summer rainfall, but more humid. And they got different responses, both to the creasing treatments they applied, and also there is comments about drop. So it could be, because remember that to me, it was unusually foggy. I didn't remember a foggy period like this for, I don't know, 15 years, 20 years maybe. But the other unusual thing about this foggy period that we had was usually when we have that thick tule fog, it's freezing, you can't stay warm. And this was warmer. I mean, it was foggy, but it was warm. And so I think that goes along with the fact that we have this very unusual weather patterns. And I would think that potentially that wet, damp weather that we had also could have played a role. But I think it's probably really the temperature. The temperature enhanced the aging of the fruit. Maybe the warm temperature also influenced the development in the abscission zone for the fruit to drop. We just really don't know. And of course, if you fund research on this, you'll be guaranteed it'll never happen again because you're funding research on it. But I think you should. I think there should be an effort to try to understand this, because I think the weather is changing, and this may be a sign of the future. The other thing I think that we need to be thinking about is that as we have warmer falls, color break may be affected. Because color break, you need the onset of low temperature. And if we're having falls that are warmer and later in terms of the cold weather coming in, we may be affecting color break. And who knows what that will do? Who knows what that would do to the impact of spraying the trees with gibberellin? Who knows what that will do in terms of degreening the fruit after harvest? I mean, it's all a big unknown.
We do have a question coming in. So, what are your thoughts on the tank mix of citrus fix along with copper, zinc, lime foliar sprays? And how much efficacy do you think is lost due to pH differences between the materials?
Well, I mean, definitely Jib likes a slightly acidic mixture. So if you're affecting, like with lime, the pH, you are, I can't give you a percentage, but you are most likely decreasing the efficacy somewhat. I'm sure if you dig into the literature, I didn't do this, but I'm sure if you dig in the literature, Dr. Coggins looked at that. There
Uh,
is a bunch of articles in old citrographs by Coggins on gibberellic acid application.
so we have a question asking, is there an organic 2,4-D product available? Also, I've noticed on my trees, there is less puff and crease on the south side of my Washington navel orange trees, and even some fruit will have puff and crease on the side away from the sun that's not facing the sun. Also, fruit less shielded by leaves seem to have less puff and crease. Is there any information on that?
Yes, that's a great question. If you read the older literature, they talk about interior fruit tending to have more puff and crease than exterior fruit. They talk about the interior fruit usually have a thinner peel, and thinner peel is usually, not always, associated with increased puff and crease. And so all that in is true. So very good observation. It's been reported and observed before.
And then for that first question, is there an organic?
Oh, they're organic? Well, 2,4-D is a synthetic compound, so I doubt very much that I'm not an authority on that. I don't think so, probably. But I could definitely be wrong. I mean, perhaps that's where applying a jasmonate compound, if there was one that could be organically approved, might help, but there's no data to back that up.
Well, while we wait to see if there's any more questions that come in, I just want to let everyone know about our next webinar that's on Tuesday, June 23rd at 10 o'clock in the morning. That'll be with our IPM entomologist, Dr. Ivan Milisavovich, and he is going to talk about our California craft program and the progress they've made in the desert and how it's expanding into Ventura County. So if you're interested in that, please visit our website to sign up. Looks like we did have one more question come in.
Yep. It's asking if one uses just gibberellic acid without the 2,4-D, would it affect the fruit drop?
It so you saw in the data Tripty didn't include that a jib only. They had a 2,4-D only and a 2,4-D jib application. But the older work that was done here in California, 2,4-D is the one that stops the fruit drop. It's not the jib. Although jib will weakly, I mean, it does have some effect, but it's very, very minor. The main effect the jib has is on the aging of the peel and delaying color break, which go hand in hand. So that goes back to that recommendation that if you're going to go through the time and expense to apply gibberellin so the peel is in better condition into January and February, the drop season typically, like for navel oranges, starts, especially in Parent Washington, will start in January and February. That's where you get a lot of fruit drops. So if you're going to go through the time and the trouble to apply the jib to keep the fruit in good condition, then you need to have a 2,4-D spray as well, so that fruit you've invested in don't drop.
Well, I don't see any more questions at the moment. We'll stay on for a few more minutes to see if there are any more questions. But thank you again to everyone who joined today. As mentioned, this webinar will be available to watch on our website, citrusresearch.org, in the next several weeks. And to stay up to date with upcoming events and research, please visit our website. Again, we'll stay on for a few more minutes if anyone has any questions, and Mary Lou's contact information is on the screen as well if you want to reach out to her directly.
I did have one more question since there's not other questions coming in. Um, you mentioned, so I guess my question is there any known, I guess not negative effects when you spray a PGR, if so you're increasing hormones in one part of the plant. Is there any research that says by increasing here, you're decreasing maybe another metabolite or anything like that at the time of the spray? Because I know you mentioned like if you spray jib um, at the wrong time, it may affect like the flowering later on down the road. But is there any literature that says by increasing jib at this sort of time, you're then decreasing another metabolite or another
Oh, yeah.
effect?
I mean, I didn't get into it because there's no time and it's really a little bit above my knowledge base. But the recent reviews go into great detail about, especially gibberella and auxins, how they affect different signals in the plant. And so it's really a balance of all these internal signals in the plant and how they affect different metabolic pathways in the plant, like on the flower induction, how Jib can decrease the expression of different signals for flowering. And so, yes, it isn't just I'm spraying Jib, and you have to think about why, when I spray Jib, why does that peel stay green? What processes in the peel is it affecting that peel stays green and it slows down, crease and puff, and et cetera, just like with the with 2,4-D. We know the fruit abscisses because there's an abscission zone on the button of right above the button of the fruit. So we think, okay, 2,4-D slows us down, but what is the mechanism of that? And in this day and age where we can look at markers and gene expression, the literature is full of discussions about this, but it's very, very complicated. I didn't have time to go through that, and I'm really probably not the one. Tripty might be a good one for you to invite to talk about that, because she's looking then like at the jasmine. She looked at the expression of these phytohormones in the peel and the abscission zone when they did that work in Florida.
Thank you.
You're welcome. I gave you a roundabout answer, sorry.
Well, good.
Well, with no more questions, we'll go ahead and end the webinar here. Again, thank you to everyone who joined us today. And if you do have any further questions for Mary Lou, you can reach out to her email, which is on the screen. And thank you
Thank you
again to
very
Mary Lou
much.
for her great presentation.
Thank you, and I'll send you that PDF.
Thank you.